Dale, play.
Escaneando.
Vale, ahora. Perfecto.
What is gene editing? Gene editing or genome editing is a process in which small intended changes in the DNA are introduced in the genome of a living organism such as plants. It also targets the changes to specific locations in the genome. This process allows to generate new plant varieties in a fast and directed manner. For example, to improve characteristics and create benefits for specific societal objectives. After years of studying chicory genetics, scientists have figured out how to develop varieties with very specific and desired features, such as increasing the production and quality of beneficial substances such as terpenes and inuline. In order to obtain these new plant varieties, we need to take a look at the genes of plants and study their function carefully. Genes define the properties of a plant and are found in a DNA, a molecule found within the cells of plants, specifically in the nucleus. In short, DNA has a helical, double-stranded shape and encodes almost everything about the plant. Changing the DNA coding allows to obtain changes in the plant's features. In order to achieve the desired changes in a plant's DNA, a protein called CRISPR-Cas9 is used to cut the double-stranded DNA at a specific location. The cut is glued together again by the plant itself but introduces small changes in the DNA that encode for the desired features of the new plant. For instance, one can use gene editing techniques to improve the amount of healthy terpenes or inuline food fibers in chicory by choosing carefully which gene to target with the CRISPR-Cas9 enzyme to make the cut in the double-stranded DNA chain. Click Start Game to begin the intended changing of the plant's DNA. Be careful, you have very limited time to perform these steps. Pick up the DNA splicing enzyme and guide it to the gene you would like to improve and see what happens. Well done! The DNA will be cut. The plant will then try to repair the DNA in the gene you directed the enzyme to. The repair has caused a small change in the DNA. Great! We can now grow a new chicory plant and see whether we have developed an improved gene.